Perfect. So I will start now with a short introduction, and then I will move to the PDH identification and its characterization, and finally its engineering to enhance this promoter, and I will conclude with the concluding remarks. So first, when we start a new bioprocess for recombinant protein production, we start generating the strain, then we move to small-scale cultivations, and then to the bioreactor scale to optimize and to characterize and optimize the process. Then sometimes we go back to the strain engineering, and the final goal is always to arrive to the pilot scale. However, most of the cultivation conditions are highly dependent on the strain constructed, because here you have to choose the promoter. As you all know, the promoter is a regulatory element that drives the gene expression, and it can be weak or strong depending on the transcriptional rate. And we can classify them into constitutive promoters if they are always active, like the PGAP promoter or these other two. And then we have regulated promoters, like those that are inducible with methanol or ethanol, and here we have the POX1 that we all know. And then we would have another class that we call derepressible. That means that when the carbon source is in excess, the promoter is inactive, and then when this carbon source is depleted, we have an activation of the promoter. And here we would have these three examples. As you see, we have methanol in different promoters, and this brings me to the next question. Do we want methanol or not in our cultivations? So I think you all know the detrimental effects that we can have in a methanol cultivation with Pichia, and also the higher operational costs that we can have. So here our decision would be that we are against methanol. However, working with inducible promoters have an advantage, because we can separate the biomass growth from the production. And this is an advantage because when we are working with growth-coupled production promoters, the profile of the cultivation looks like this. So the biomass and the protein concentration increases together, and then when we arrive to the maximal biomass concentration, we have to stop the cultivation. On the other hand, when we have a growth-decoupled production, we can have first the biomass growing phase and then the production phase. So we can extend the cultivation and even produce toxic proteins that with a growth-coupled approach would be quite difficult. So with all this information, if we could choose a promoter, our ideal promoter, it would be strong, methanol-free, and growth-decoupled. And our aim would be to have also an induction based on carbon-restrictive conditions. So to achieve this, at BC they perform a shake flash cultivation with these carbon-restrictive conditions, because they wanted to study if there were overexpressed genes under these conditions. So they performed RNA-seq, and they saw that HSP12 showed the maximum expression levels. And when we checked in the literature, we saw that this gene encodes for a heat shock protein 9 or 12 in Saccharomyces. And this gene is highly induced under different stressing conditions, including the starving conditions that we were looking for. And in Saccharomyces, the promoter of this gene has already used to drive the GFP expression and use it as a general stress reporter. And in Pichia pastoris, the promoter of this gene was already recommended for recombinant protein production in 2018. So with all this information, we move forward and we isolate the promoter, and to do so we systematic truncated the 1000 base pair sequence upstream of the gene in different truncations. And these truncations were evaluated producing GFP. And the aim here was to find the the minimum length with the maximal strength. That was this transition here that we called PDH. Then we wanted to study the performance of this promoter producing a relevant enzyme that was industrial relevant enzyme that was CalV, the lipase B from Candida Antarctica. And we constructed then this new strain. We performed screenings to select a clone with neutral integration, and then we got PDHC, that was a single copy clone. We studied the performance of this strain first in shake-fast cultivations under carbon restrictive conditions. To do so, we started with a batch phase, and then we added, when the glycerol was depleted, we added feed pits to the cultivation. So they are glycerol discs that release glycerol at a slow feeding rate, achieving these pseudo-starving conditions. And we studied the PDHC strain compared to the benchmarks GAPC and UVPC that are constitutive, and PDFC that is irrepressible. So here we have the mean optical density of the 4 cultivations. You can see here first the batch phase and then the pseudo-starving. And here you can see the lipolytic activity. And regarding PDHC, you can see that at the beginning, during the batch phase, there's no production, and the production boosted after the pseudo-starving phase was applied. Conditions were applied, therefore showing this growth decoupled profile. And considering these good results, we moved forward to the bioreactor, and the final titer was 5.8 higher than with GAPC. So then we moved to the bioreactor, and here we performed carbon-limiting fed-batch with PDHC at low Mo, and we compared the results with GAPC at low Mo and fast Mo. Here we have the results in terms of biomass concentration and CalV titer, and you can see that PDHC outperformed GAPC 1.4 times higher in CalV titer and 2.3 at low Mo and fast Mo. Considering these results and considering that this promoter is growth decoupled, we wanted to test if growth decoupled approach worked in bioreactor. So we developed this new approach that we call pseudo-starving. So after a batch phase, we started a fed batch phase at a growth Mo to a goal biomass that was 70 grams per liter. And then we applied this pseudo-starving phase, just decreasing the growth rate to a really low and constant feeding rate. Here, we are comparing the results with the PDHC, as I showed you before. This is the biomass concentration and the CalV titer. You can see how we achieved this profile. And in this case, the CalV titer was equally in both cultivations, but we achieved it earlier. So the productivities are higher. And in this case, again, you can see that during the biomass growth phase, there was almost no production, and then the production boosted, showing again this growth decoupled profile. All these results and some others were published last year in this paper in the Frontiers in Bioengineering and Biotechnology journal. And now I will explain the promoter engineering that we will submit soon to the microbial cell factories. So if we come back to our ideal promoter, we will see that PDH is indeed methanol-free and growth decoupled, and also inducible under carbon-restrictive conditions. However, we thought that it wasn't that strong as we wanted, so we thought that it was a good candidate to mutate, to enhance its strength. So we generated 2 libraries of variants, B library and A library. In the B library, we chose this sequence, this 10 base pair sequence, and the approach was to replace the first 10 base pairs of the promoter sequence by this sequence, obtaining B1, and then the next 10 base pairs also we replace it with this sequence, obtaining B2, and we did the same approach through the whole promoter, obtaining in total 35 variants. For the A library, we followed the same approach, but replacing with a triplet of adenines, obtaining 117 variants. So in total, 132 clones were obtained. These clones the performance of these clones was assessed producing GFP in big ball plates, and here you can see the fluorescence, the relative fluorescence of the V library and the A library. The best variants showed about 1.5 more fluorescence than the wild-type promoter, and from here we selected the most promising mutations, and we combined them in generating new strains, new variants. And again, we addressed the performance in deep well plate, producing GFP. And this time we saw that the variants outperformed the wild-type promoter by 2.5 times more, being all one, multivariant 2, and multivariant 2 that are this here. So, considering the results, we decided to go again with CalV and see what happens. So here I'm showing the variants that we selected, that we constructed again with CalV, some single variants and also the best multivariants. And here I'm showing already the results in shake flask cultivation under restrictive conditions. And you can see here that multivariant 2, 1 and 2 again showed the best results. And in this case, multivariant 2 outperformed by 1.9 more CalV titer, enzymatic activity. That was this mutant here. Then we decided to validate these results in the bioreactor again. And in this case, we performed again carbon-limiting fed beds cultivation at low Mo, this one here with multivariant C. And as you can see, the final titer was 1.8 times higher than with PDHC, so it was consistent with the results that we obtained in shake flask. And if we take a look to the key parameters here, you can see that multivariant 2 showed the best performance, since the titer and productive biomass yield was the higher compared with all the other cultivations. And in particular, it was almost 4 times and 2 times better than with GAP-C and PDHC under the same conditions. It's true that if we compare the productivities, GAP-C at a high Mu was better, but this is because the cultivation was shorter, and we have to consider that the titer was lower. And also when we perform these high Mu cultivations, the oxygen requirements and the heat production is also higher. So in overall, the multivariant 2C was the best strain here and the best cultivation. So to finish, I just want to say that here we identified this PDH promoter that we can say it's growth decoupled, methanol-free, and also strong. We developed a new approach for growth decoupled recombinant protein production, that is the so-called pseudo-starving condition. And finally, that generating these new PDH variants, we showed promising candidates or stronger candidates for recombinant protein production, achieving up to 2 times more final activity. I want to finish saying thank you to ACIP for funding this project, and also to my UAV team, especially to Miguel Angel, for doing the PDHC strain, and also to my supervisors, Paco and Xavi. And also thanks to the BC team, Tommy, and especially Kathy, for the great work that she did with generating all the variants. And thank you.
How repressed are these promoters when you're actually working with them in terms of doing transformations and working on plates? So we certainly have run into a fair number of proteins that are like toxic on a constitutive promoter, and you can't even get transformants. So I was wondering whether they're repressed enough when they're growing on a plate to not express the protein.
Yeah, I think we didn't check this. I didn't do all the cloning work, but I would say, at least from literature, from Pichia and also Saccharomyces, apparently the PDH is highly repressed on glucose and on glycerol. So I would say that on plates also. So we have seen in liquid plates and shake flask.
Can comment on this from the early days. So it's repressed, but not. You can't compare anything, I think, to AOX. Yeah.
You had shown in the beginning that the native PDF promoter outperforms the native PDH promoter. Did you also benchmark the mutant versions for the PDH you had generated against the PDF?
Yeah, I wasn't showing here, but yeah, PDF still outperformed PDH mutants. But yeah, I think it was, so PDF was about 5 times better than PDH. And compared to the multivariant 2, it was about 3 times higher, or less than, no, sorry, about 2. You will see in the paper that we will submit soon.
Very much looking forward, thanks.
I have a question about impurities. Do you have any idea how these different promoter systems and modifications impact the downstream processing? Would you say it's mostly strain dependent, or do these different manipulations of the expression system impact the downstream processing and impurity profile?
We ran some SDS pages and we didn't see any difference in the protein pattern, and in fact the purity was already quite high in all. No, we didn't see any difference.
That's another discussion I would like to initiate in even other impurities. Most times we don't care. We look on SDS gels, but if you produce on large scale, there are other things which are mostly not discussed. It would be nice evening.
Yeah, one question over here. Thanks for your nice talk. Can you explain to me what you mean with pseudo-starvation? What does that exactly mean?
Yeah, of course. So the cultivation is not growing. You can see in the biomass concentration that was constant, but we were feeding them. So all the feed was for maintenance. And yeah, we didn't want to call it starving because we did feed them, but it wasn't growing. So yeah, we came up with this name. But basically, it's in the bioreactor level, we were feeding at the same rate as the glycerol feed bits from the shake flask. We were feeding the same rate as the glycerol feed bits.
So you did generate data to figure out your maintenance coefficient and then calculate your feed rates back. Is that what you did?
That's
okay,
it.
Gotcha. Thank you.
Do you overexpress any chaperone along with this promoter, or is that just the promoter itself that you are looking at?
If we overexpress...
Any chaperones are overexpressed in
No, just the promoter. So here we just evaluated the promoter's strength. So all strengths were the same, just the promoter changed.
Thank you.
No helper, no nothing.